Welcome to the webinar presentation of group number 2. The theme of the webinar is drug recall cases, and we will now present the drug rotacocaine alpha activated. What are we going to discuss in this presentation? Firstly, what is rotacocaine alpha and what is it intended to treat? What was the adverse drug reaction and why was it not predicted? And finally, how was the problem resolved? Rotacocaine alpha is the activated form of human protein C, and it is synthesized by recombinant DNA technology. It is a 55 kilodalton-sized glycoprotein, and it belongs to serine proteases. The product was introduced under the market name Sigris by Eli Lilly and Company, and it was approved by the Food and Drug Administration in 2001. The European Commission granted Sigris a marketing authorization valid through the European Union a year later. Its intended use was the treatment of adult patients suffering from severe sepsis to reduce the mortality. Sepsis is a serious, life-threatening condition that arises when body's response to an infection is causing damage in its own tissues and organs. In severe sepsis, the inflammatory procoagulant and fibrinolytic effects can lead to shock, organ dysfunction, organ failure, and ultimately to death. Protein C has been shown to be an important prognostic indicator in patients with sepsis. Decreased protein C levels have been linked to mortality in sepsis and septic shock. The specific mechanism of Sigris in the treatment of sepsis is not completely understood, but natural activated protein C acts as an anticoagulant and has antithrombotic, antifibrinolytic, and anti-inflammatory effects. Excessive blood clotting is a problem during severe sepsis because blood clots can block the blood supply to important organs such as the kidney and the lungs. Since Sigris is an anticoagulant, it is pretty self-explanatory that it affects the blood coagulation potential in the host. Sigris was noted to cause an increased incidence of adverse bleeding events when it was compared with placebo. This effect was mostly limited to the duration of the drug infusion. In the recombinant human activated protein C worldwide evaluation in severe sepsis, or PROVIS study, the incidence of bleeding with Sigris was 3.5% versus 2% for placebo. This PROVIS trial consisted of 1,690 patients, and the study showed a 28-day mortality rate of 24.7% in patients treated with Sigris in comparison to 30.8% in the placebo group. This would mean an absolute risk reduction of 6.1% or a relative reduction of 19.4%. Here you can see some more numbers from this placebo study, so feel free to pause the presentation if you are interested in seeing them in detail. Sigris had a promising start in the year 2000, and the PROVIS phase 3 trial was actually stopped early because of the drug's high efficacy, reducing the relative risk of death by almost 20% when compared with placebo. As we just showed, the increased risk of bleeding had actually been noted in the trials, but then the patients at an increased risk of bleeding were excluded halfway into the trial. The pediatric clinical trial was also stopped after the results with 477 patients between 0 and 17 years old who had received the treatment did not prove the efficacy of Sigris. The study showed a higher rate of central nervous system bleeding in Sigris versus the placebo group, and Sigris was therefore contraindicated in children below the age of 18. Despite of the increased occurrence of the bleeding, the trials had also demonstrated that fewer patients treated with Sigris had died than patients treated with placebo, leaving the authorities pretty puzzled. Because the fact is, even though many investigational drugs had been proposed as treatment for sepsis in the past, only Sigris had shown any survival benefit. So when Sigris was approved by the FDA in 2001, it was the very first and thus the only approved treatment for severe sepsis with no alternative medication to choose from. After the first data about Sigris had been published in 2001, the United States Food and Drug Administration advisory meeting experts raised several concerns, including worries about why the Eli Lilly changed the enrollment criteria in the trial, and they were faced with a lack of therapeutics on one hand and not enough clinical data on the other. So 10 panelists were voting to approve the drug and 10 were voting to reject it. In an attempt to follow some of the criticism, the approval was restricted to a subgroup of the PROVIS study population. And in the following year, the European Medicines Agency Committee for the Medicinal Products of Human Use was estimating that Sigris's benefits are greater than its risks and was recommending that it would be given marketing authorization. The same year, Sigris was authorized in EU under exceptional circumstances. This means that at the time of the marketing authorization, the applicant was not able to provide comprehensive evidence on the efficacy and safety of the medicine, and the benefit-risk balance of the product has to be reviewed regularly. So every year, the European Medicines Agency would review any new information and give updates as necessary. And in the EU, the use of Sigris was restricted to the most severe sepsis patients with at least 2 organ failures, and the treatment had to be started within 48 hours of the onset of severe sepsis. Sigris was also contraindicated in children below the age of 18 and in the patients that are at increased risk of bleeding.
After the annual reassessment in 2007, the European Medicines Agency concluded that the initial efficacy results of the original PROVIS study have not been reproduced in further studies, and thus more clarification is needed about the benefit-risk balance of Sigris. Following EMA's request, Eli Lilly started conducting a post-marketing placebo-controlled trial called PROVIS Shock. The 2008 revision of the Surviving Sepsis Campaign guidelines downgraded the evidence provided by the original PROVIS trial to those of an upgraded observational study, recognizing its limits. In 2009, the FDA added to the prescribing information for Sigris description about the increased risk of bleeding and included a statement in the warnings and precautions section that bleeding is the most common serious adverse reaction experienced by patients who receive the drug. The warnings and precautions section also listed a number of risk factors for the increased risk of bleeding that should be taken into account when considering use of Sigris therapy. The contraindications section stated that Sigris is not to be used in the clinical situations where bleeding could lead to significant morbidity or death. Furthermore, the FDA was working with the manufacturer to further evaluate the incidence of serious bleeding events and mortality in patients who received Sigris. The FDA urged both healthcare professionals and patients to report side effects from the use of Sigris to the FDA's MedWatch Adverse Event Reporting Program. And in 2011, Eli Lilly released the results of the post-marketing trial. In PROVIS Shock trial, Sigris failed to meet the primary endpoint of a statistically significant reduction in mortality in patients suffering from severe sepsis. In this trial of 1,696 patients, 851 patients were enrolled in the Sigris arm, and 845 patients were enrolled in the placebo arm. Results based on preliminary analysis done by Eli Lilly and Company that were submitted to the FDA showed a 28-day all-cause mortality rate of 26.4% in Sigris-treated patients compared to 24.2% in placebo-treated patients. The small difference in the mortality is not statistically significant. Curiously, the risk of severe bleeding events was 1.2% in the Sigris arm and 1.0% in the placebo arm, suggesting there was no increased harm, while it was increased in the Sigris group in the 2001 PROVIS trial. These results called into question the overall benefit-risk balance of Sigris. Here we have listed some pro and contra thoughts about Sigris and what we know about it at this point. Sepsis has a high chance of death, from 30% to 50% occurring in severe sepsis and as high as 80% in septic shock. And since there are no other survival rate increasing drugs in the market besides Sigris, it could possibly be the patient's only hope. The first trial did show a mortality reduction of 20% compared to placebo, while later studies then contradicted this. The product was controversial from the start and barely got the marketing authorization. Eli Lilly had been hoping for yearly sales of over $1 billion, but after 5 years in the market, the total sales had only reached $200 million, which was a huge financial disadvantage when looking at it from the company's perspective. So what are the consequences of the new information from the PROA shot? The manufacturer Eli Lilly and Company announced a worldwide voluntary market withdrawal of Xigris in October 2011, nearly 10 years after the drug was initially approved. Eli Lilly also decided to discontinue all ongoing clinical trials. On October 25th, both the FDA as well as the EMA gave a statement instructing the healthcare professionals that Xigris treatment should not be started in new patients. Xigris treatment should be stopped in patients being treated with Xigris. All remaining Xigris products should be returned to the supplier from whom it was purchased. The withdrawal of Xigris marks a U-turn in the treatment of severe sepsis, which is plagued by starts, stops, and confusion. Since the withdrawal of Xigris, no new drug for sepsis has reached the market. Speaking of market and marketing, the drug company Eli Lilly and Company has been receiving criticism about their marketing tactics. A recent New England Journal of Medicine article blames Eli Lilly for overzealous promotion of Xigris. According to the newspaper Boston Globe, Eli Lilly and Company funded medical guidelines created for the treatment of sepsis in an effort to boost sales of a drug with questionable benefits. The allegation was made by senior scientists at the National Institutes of Health. They said Lilly tried to shape the guidelines for use of the drug Xigris by sponsoring a three-pronged marketing campaign. The first two phases are by now almost standard practice in the industry. Lilly paid a task force to spread the word that hospitals were rationing Xigris because of its cost, which forced doctors to decide who would live and who would die. Lilly orchestrated the development of practice guidelines to treat sepsis that called for early use of Xigris. And Lilly is sponsoring lobbying efforts to turn the guidelines into quality standards. Hospitals that follow such quality measures receive higher payment from insurers. So what's happening here? Basically, an influential group of doctors are being lazy and greedy, and Lilly is enabling their behavior. The outcome of this is that the public are paying for higher product sales and receive suboptimal healthcare in return. Another matter what we have learned from the case of Xigris is that post-marketing drug studies are important in order to test efficacy and safety in a broader population. That will give a wider look of how it affects the bigger mass, which is something that's not possible to do in clinical trials due to the relatively small groups with possibly little diversity. To conclude, Drotrosogin alfa was introduced in 2001. It was however withdrawn after 10 years in the market due to two reasons. Firstly, there was evidence about a serious adverse drug reaction in the form of increased bleeding. Secondly, it was shown, following study trials, that the drug had no significant efficacy in the mortality reduction in patients with severe sepsis. At the time, Drotrosogin alfa was the only approved drug for the treatment of sepsis, and until this day, no similar or efficient drug for sepsis has been developed. Namely, the development of a new treatment for sepsis has come to be a slow and a complex progress. Finally, thank you for your attention and feel free to ask any questions and leave comments in the discussion forum.